Hi, this is Lloyd. I'm a bioinformatician at the Davis Research Center, University of Adelaide. Today, I'm going to share with you some lessons learned from assembling high-quality reference genomes for domesticated and vulnerable ruminants. Here are the ruminant species that I'm currently working on: the water buffalo, gaur, Angus, and Brahman cattle. Water buffalo and cattle are livestock animals valued for their milk and meat. Gaur is the largest species of wild cattle that may become endangered in the near future. It is native to South and Southeast Asia and has been listed as vulnerable on the International Union for Conservation of Nature Red List. If you would like to know more or participate in this area of work, please consider the Bovine Pangenome Consortium, or BPC. I'm a member of this consortium. My interest aligns well with the BPC, as one of my research goals is to create reference genomes of multiple bovine species to facilitate genetics, evolution, gene expression, traits improvement, and other studies. To give you an example of what interesting biology we can study with high-quality reference genome, I will use the Brahman and Angus cattle. There are two subspecies of cattle: the Bostoros taurus, which is represented by Angus here, and the Bostoros indicus, which is represented by Brahman here. Angus and Brahman are the names of cattle breed. Taurine cattle are adapted to temperate climate, has no hump, more fertile, but less tolerant to heat and ticks. Indocene cattle are adapted to tropical climate, has a hump, less fertile, but more tolerant to ticks and heat. Being subspecies, one would expect their genomes to be very similar. Comparison of taurine and Indocene cattle genetic differences is a key to unlocking the secret of bovine adaptation to a changing environment. Now I'm going to touch on something more technical on how to produce high quality assemblies. This table summarizes the sequencing detail. First, you will need to collect some material from the animal. Usually we would like to sequence a female animal to avoid dealing with the problem of the Y chromosome, which is very repetitive and hard to assemble. Blood and lung are good samples to sequence. If you can, fetal lung may even be better, as contamination from bacteria should be minimal in this case. We have used both the P6 and C4 and SQL sequencing kit as the sequencing chemistry, and then SmartBell template preparation as usual. The instrument that we have used are the RS2 and SQL, with most of the data coming from the SQL machine. I wish to acknowledge Tim and Sarah for this part of the project. For assembly, we have done a few using a few types of assembler technologies. For the water buffalo, we have used the Falcon Unzip. Angus and Brahman cattle, we have used the trio binning assembly strategy with Canoe, and the gaur is just the normal canoe. For scaffolding, in the case of the water buffalo, we have used Chicago and high C reeds. For the Angus and Brahman cattle, we have used high C and bio-nano optical maps. For the gaur, it's just the high C reeds. For gap filling, we use PB jelly for the water buffalo and cattle. We didn't use any for the gaur because not many gaps were filled in the case of the gaur. Then polishing using arrow and pylon. Except in the case of the cattle, we didn't polish with short reeds because it was not necessary. The genome size for all these animals is about 2.7 billion bases. You can read more about the detail of these two work, the water buffalo and the Angus and Brahman, using these links. The gaur work is not published yet. For diploid species, there are 3 types of assemblies that one can make. Most of the assemblies in the public databases are unphased, which means the haplotypes are not resolved. The gaur assembly represents this type of assembly. Some of the assemblies are partially resolved, such as the water buffalo assembly. In this case, the haplotype is locally resolved, but phasing is not maintained across the genome. For example, over here, there is a haplotype switch. Falcon Unzip will generate this type of assembly. If you have high C data and provided there is sufficient differences in the 2 haplotypes, you can try to use a new tool, Falcon Phase, to get a more complete phasing of the 2 assemblies. The goal of diploid assemblies should be complete phasing of the 2 haplotypes, and this is represented by the Angus and Brahman assemblies. I will go into the detail of trio binning assembly as the method is still relatively new and many people may not be familiar with it. For trio binning, ideally we start with 2 genomes that are very different, but yet they can still produce a hybrid animal. In this case here, we have 2 subspecies diverge at about 1% at the genome level, and I will show you how to assemble the F1 hybrid. This method is as follows. It starts by sequencing the trio, Angus and Brahman parents, and the F1 hybrid. The F1 is sequenced with PacBio to 136 times coverage, so that for each haplotype we have sequenced more than 60 times coverage. The parent Angus and Brahman were sequenced with Illumina short reads to about 60x coverage each. All the K-mer, specifically 21-mer in this case, in the Illumina reads were identified, and the Angus specific and the Brahman specific K-mers were used to guide each and every of these PacBio reads to either the Angus bin or the Brahman bin. With this method, we group the PacBio reads to Angus, Brahman, and some cannot be assigned. The unassigned reads is about 1%, and they cannot be assigned because they are too short to contain sufficient haplospecific KMA. The mean read length is only 1.3 kb in the unassigned group, but you can see that for those that we can assign, the mean read length is more than 11 kb. We can assemble the Angus reads to give the Angus contigs, and likewise for the Brahman. Contig assembly alone is not enough in this case, and we still need scaffolding of the contigs. We use Hi-C for long-range scaffolding and the BioNano optical map for short-range scaffolding. I will not go into the detail here, but basically, optical map is crucial for resolving some of the wrong chimeric contigs, and it is also very good at ordering and orientating the scaffolds. Hi-C is very good for joining contigs to give chromosome-sized scaffolds. When I have the final assemblies, I will usually check it in a few ways, and I will only touch on two aspects here. For more detail, please refer to the links to the papers. The number of gaps tell me how fragmented the assembly is. The smaller the number of gaps, the better the assembly is. Using this metric, the recent cattle and water buffalo assemblies appear better than the human assembly, but the human assembly is more complete in the sense that it extends to include the telomeres. The gaur is not a bad assembly with 2000 plus gaps, but much more fragmented. And I think one of the reasons could be the poorer quality of the source DNA for sequencing. In the assemblies that we did, the sex chromosome are still the hardest to assemble due to the presence of repeats that break up the contigs. Using long reads, one should expect better resolution of repeats. I listed here 3 major classes of repeats that are usually found in cattle. The UMD 3.1.1 is a short read assembly of dominant, the name of a cow, and ARS UCD is the long read assembly of the same animal. UMD 3.1.1 is actually made up of more than 2 alleles from dominant, but I will not delve into this aspect. We can see that the long reads assembly is able to resolve the longer L1 line repeats and the satellite and centromeric repeats. You can see gaur show similar repeat resolution. And although I didn't show the Angus, Brahman, and the water buffalo, they all show similar results, which is longer repeats are resolved. The correspondence of homologous chromosome to a high-quality reference is another way to check and compare genome assemblies. Here's a circos plot that shows the gaur chromosome match what is expected when compared to cattle. For example, the gaur chromosome 1 is known to be made up of the fusion of chromosome 2 and 28 in cattle. And we do see that in our assembly. Yet another way to check is to look at the assembly of difficult, polymorphic, and repeat-rich region, such as the MHC II. The UMD 3.1.1, the short read based assembly, is not good at assembling this region. As you can see that there are many gaps in this region. The long read assembly version of the same animal, ARS UCD 1.2, is much better in this regard. The Brahman, water buffalo, and human have very good assembly of the MHC II region. The gaur is not too bad in this regard, but some of the MHC II genes are placed on a different chromosome, possibly an assembly error. As you can appreciate, we need high-quality assembly to study the evolution of this important cluster of immune genes. One of the most diverse genomic regions between the Brahman and Angus cattle was observed on chromosome 15. A region of about 1.4 billion bases has 3 copies of a gene known as fatty acid desaturase 2 genes, FADS2P1. 3 copies in Brahman, you can see here, and 2 copies in Angus. In both the Brahman and Angus, these FADS2P1 genes are encoded by 10 to 12 exons, and the entire region is assembled without gaps. So we can be confident that this diverse region that we see here is not because of gap. We have identified 16 amino acids residues under positive selection for this gene, 10 of which are located in a small exon 7 of only 60 bases. We hypothesized Bos indicus has 3 copies of these FADS2P1 genes to regulate the composition of fatty acids that constitute the cell membranes and could alter water permeability and heat loss from skin. The availability of high-quality assemblies enable us to compare gene families to find those that are changing in gene number more than expected. Here we have used genomes annotated by NCBI and have counted N50 more than 20 million bases. We found significant changes in gene families involved in protein degradation, olfactory receptor, detoxification, and immune system. I will now talk about a different type of long-read sequencing known as Iso-Seq, which are useful for annotating transcripts and phasing transcripts. In our typical genome assembly project, we will generate RNA-seq reads from various tissues to guide the genome annotation process. In the Brahman and Angus project, we have also generated Iso-Seq reads from the brain, heart, muscle, kidney, liver, lungs, skeletal muscle, and placenta tissue of the F1 hybrid. So this is the offspring from the Angus and Brahman cross. The Iso-Seq, so what we do is We have the Iso-Seq SmartBells libraries created according to the PacBio protocols. And briefly, two size-selected cDNA pools were created, one with an average cDNA size of about 3 kb and the second one with a size of about 7 kb. We combine the two pools and proceed to SmartBell sequencing. When we get the Iso-Seq data, we process it using the Iso-Seq 3.1 version 3.1 pipeline. The process is as follows. We start by generating the circular consensus sequencing reads, or the CCS reads, classify them to full-length non-concatenator reads based on these criteria, and then we will cluster these FLNC reads to each isoform and then polish them to create high-quality full-length transcripts. Next, we use a tool known as SCANT2 to characterize the transcript annotation. 83% of the Iso-Seq transcripts fell into coding region, which is expected. Here's the histogram of transcript length distribution, where we can see a peak count at transcript length of about 4 kb. We validated the SNPs called using this tool known as Unphased, which use the Iso-Seq data, with SNPs called from RNA-seq. So these are RNA-seq data of the brain, liver, lung, muscle, and placenta of the same F1 hybrid. And we also use the SNPs. Well, in this case, all the SNPs called will be the Angus SNP, because we map our Iso-Seq data to the Brahman genome. So calling SNPs from the short reads, just the whole genome sequencing short reads, basically we're calling all the Angus SNPs. The concordance between the three different ways to call SNPs is very high, which is at about 87%. Here's the Wiley plot of the proportion of Brahman allele, which was calculated as the normalized count of Brahman alleles divided by the sum of the count of both Brahman and Angus allele. Transcripts showing allelic imbalance will, with a higher expression in Brahman, have values closer to 1, whereas those with higher expression in Angus will have values closer to 0. We found some genes with interesting allelic imbalance, such as this gene, ARIH2. This gene is the most highly expressed Angus gene in the brain, and you can see it displays allelic imbalance across a few tissues. Without Iso-Seq, it is hard to study the allelic imbalance at the transcript level. Here's a plot to show the complexity of the transcript expression of this gene in different tissue. A denotes Angus and B denotes Brahman. The previous Iso-Seq experiment is more on using Iso-Seq for annotation and to begin testing its ability to phase transcripts. However, it was done with only one replicate per tissue. This does not allow us to perform any statistical testing. We have recently set up an experiment to compare transcript quantification at the gene and transcript level. We have used 3 biological replicates of female liver samples of Angus and compare it to 3 biological replicates of female liver samples of Brahman. We did 2 SMART cells per individual. This was done using the latest SMART cells that have much higher throughput. We also have RNA-seq data for the same individuals. This allows us to compare gene expression and quantification using these 2 different technologies. We use the same Iso-Seq pipeline to get full-length transcripts. Our initial experiments showed some variation in the number of genes and transcripts found per individual. Next, we will try to quantify gene expression differences and compare with the RNA-seq results of the same animals. I do not have the results yet, so stay tuned for this development. So in closing, intraspecific crosses such as the Brahman and Angus subspecies cross provided considerable variation to enable haplotype resolve assemblies. Long PacBio reads are useful to span long repeats to preserve assembly contiguity. Iso-Seq reads are useful for genome annotation, phasing transcript, and maybe even transcript quantification. Many people are involved in this project, and I wish to thank these individuals in particular for their help in the project so far. Thank you.